Carbohydrate biomolecule. Overview of biomolecules. Biomolecules are organic molecules that are essential for life. They are naturally occurring substances in living organisms and play key role in structure, functional and biological processes. Different types of biomolecules include carbohydrates, proteins, lipids and nucleic acids. The carbohydrates provide energy and structural support. Proteins catalyzes structure and transport and cell signaling. Lipids store energy, insulation and cell membrane structure. The nucleic acids store and transmit genetic information. Importance of biomolecules. They are involved in vital functions like metabolism, growth, reproduction and maintaining homeostasis. Carbohydrates. Carbohydrates are good sources of energy. Carbohydrates are long chains of sugar. They have aldehyde or ketone derivatives of polyhydroxy alcohols or compounds which give these derivatives on hydrolysis. Carbohydrate metabolism is a process by which the molecules break down carbohydrates to produce energy and stores them when not needed. Importance of carbohydrates. The carbohydrates are most important energy sources of living organisms. They are also known as energy giving foods. Why carbohydrates are important? They are the main source of energy. The carbohydrates are broken down into glucose, which gives energy to the body and brain to work properly. The carbohydrates are fuel for cells. That is, every cell in the body uses carbohydrate as a main source of energy. That is, for example, glucose. Structural role. That is, in plants, the carbohydrates like cellulose give strength to the cell walls and help maintain plant structure. Carbohydrates store energy. That is, extra glucose is stored in the liver and muscles as glycogen for later use. Structure of carbohydrates. The carbohydrates are made up of smaller units called monosaccharides, also known as simple sugars. These are most basic forms of sugar. Important monosaccharides include glucose, which are found in blood, sugar and fruits. Fructose, found in fruits and honey. The bonding in carbohydrates is done by glycosidic linkage. When two monosaccharides bond, they form a glycosidic linkage. This happens through a dehydration reaction where a water molecule is removed from the reaction. For example, glucose plus glucose gives maltose, plus the byproduct is water. Types of carbohydrates. There are 4 different types of carbohydrates. Monosaccharides, which are simple sugars. These are the basic units of carbohydrates and cannot be broken down further. They are soluble and sweet in taste. Example, glucose and fructose. Second, disaccharides. Double sugars. They are double sugars made up of 2 monosaccharides joined by glycosidic bond. They are soluble in water and sweet. Example, sucrose, lactose and maltose. Oligosaccharides. They give 3 to 6 monosaccharides units on hydrolysis. Example, raffinose and stachyose. Polysaccharides are the complex carbohydrates. It is a long chain of many monosaccharides. Polysaccharides are soluble in water. They are not soluble in water. They are used for storage and structural purpose. Example, glycogen, cellulose. Metabolism activity of carbohydrates include metabolism, that is, glucose is broken in the form of ATP, used as a body's energy source. Cellular processes used in cell signaling, division and building cell walls. Genetic information, that is the sugars from the backbone of DNA and RNA. Energy storage, stored as glycogen in animals and starch in plants. The metabolic activities of carbohydrate includes glycolysis, link reaction, Krebs cycle and electron transport chain. The carbohydrates, that is primarily glucose, undergo oxidative degradation to provide energy in the form of ATP through a series of biochemical processes. Glycolysis. Glycolysis occurs in the cytoplasm of a cell. It is a anaerobic type of process that which does not require O2. Uh, in glycolysis, glucose is broken down into pyruvate molecules, yielding ATP and NADH. The enzymes involved in glycolysis are hexokinase, phosphofructokinase, and pyruvate kinase. Link reaction. It occurs in the mitochondrial matrix of a cell. The enzymes uh, used are pyruvate dehydrogenase complex. In link reaction, the pyruvate is converted into acetyl CoA with the release of CO2 and production of NADH. Krebs cycle, also known as TCA cycle, it occurs in the mitochondrial matrix of a cell. It is a aerobic process which indirectly depends on the oxygen. In Krebs cycle, the acetyl CoA is oxidized into CO2, producing NADH, FADH2, and ATP in a cyclic pathway. Main enzymes in Krebs cycle include citrate synthase and alpha ketoglutarate dehydrogenase. Uh, Electron transport chain, also known as ETC cycle, it occurs in the inner mitochondrial membrane of a cell. It is a aerobic type of reaction. In ETC, electrons from NADH and FADH2 generate a proton gradient used to synthesize ATP and H2O. Oxidation of one glucose molecule can give approximately 36 to 38 ATP molecules, which are further used for energy. Techniques used to study carbohydrates. Uh, First, chromatography, which is separation of sugars. Colorimetric assays, that is quantification of carbohydrates. UV spectrophotometry, that is quantification based on absorbance of carbohydrates in a solution. Centrifugation, that is cell isolation of of carbohydrates from a solution. Electrophoresis, that is analysis of glycoproteins from a solution.